Namaskar. Today we will study about the ribonucleic acid. So ribonucleic acid, it is a nucleic acid. There are two types of nucleic acid. One is deoxyribonucleic acid and the other is ribonucleic acid. And nucleic acid, which is a polymer of nucleotides, and nucleotide consists of a sugar, a base and a phosphate. And in RNA or ribonucleic acid, the sugar is ribose sugar. In DNA, the sugar is deoxyribose sugar. This is the difference. And similarly, the bases, there are two purine bases and three pyrimidine bases. Out of these bases, three are common to adenine, guanine and cytosine are common to both DNA and RNA. But uracil is present only in RNA. In DNA, thymine is present. So in RNA, the bases are adenine, guanine, the two purine bases, and cytosine and uracil, the two pyrimidine bases. And the sugar for RNA is the ribose sugar. This is the ribose sugar. The nomenclature starts with 1 dash, 2 dash, 3 dash, 4 dash, and 5 dash. This is the fifth carbon. And this ribose sugar, when binds to a base, it forms the nucleoside. Here in this example, we have the adenine, the base, the sugar is the pentose sugar, and this base plus sugar is the nucleoside. So here the base is adenine, so the nucleoside is adenosine. And then with a phosphate, it becomes adenosine monophosphate, 2 phosphates, adenosine diphosphate, and with 3 phosphates, it becomes adenosine triphosphate. So AMP, ADP, ATP, all are the examples of the nucleotides. Then a chain of the nucleotides. For example, nucleotides will be your say adenosine monophosphate. Then similarly, guanine nucleoside is guanosine and the nucleotide is guanosine monophosphate. Similarly, cytidine monophosphate and uridine monophosphate. These four, adenosine monophosphate, guanosine monophosphate, cytidine monophosphate and uridine monophosphate. These four are when joined to form a polymer, it becomes RNA. So here you can see RNA is mostly a single-stranded molecule where you can see sugar, phosphate, a chain is there, and to each sugar, one of these bases, adenine or uracil or cytosine or guanine, are linked. So this is 5 dash direction and this is 3 dash direction. Means in this molecule, in this ribose sugar, the 3 dash position and 5 dash position, and the chain can be there, and the phosphate can be attached to either this 5 dash position or to the 3 dash position. And usually what happens, the phosphate is linked to 5 dash position of one sugar and the 3 dash position of another sugar. So here you can see this 5 dash position is linked to one phosphate, this 3 dash phosphate is linked to one phosphate. And in other words, you can say the phosphate is attached to the 5 dash position of one sugar and the 3 dash position of the other sugar. And in this direction, this 5 dash to 3 dash direction, the chain is elongated. And we can have RNA. And how this RNA is synthesized, we will discuss. Before that, mostly this RNA is single-stranded, but it can also be double-stranded in some virus, and there is one RNA, small interfering RNA, that can also be double-stranded RNA. And there is also circular RNA, and which has functions like it can be involved in transcription, in alternative splicing, and the one major function of the circular RNA is it inhibits the action of microRNA. So let us first discuss this. What is this? What is the types of RNA? So the RNA can be two types: coding RNA and non-coding RNA. Coding RNA means it has the codes which are it has the information in the form of codes which is present with the DNA. So the example is your messenger RNA, and most commonly we know these three RNA: messenger RNA, transfer RNA, and ribosomal RNA. So now we can classify RNA into coding and non-coding. Only in the protein synthesis they provide the code which is present with the DNA. That's why this is coding RNA, and the rest are the non-coding RNA. They have nothing to do with the codes, genetic codes provided for protein synthesis. And these non-coding RNA can be divided into housekeeping and regulatory. And we know the transfer RNA and the ribosomal RNA, they are also involved in protein synthesis. Transfer RNA helps in translation. So after the transcription, transcription is the process in which DNA is converted to messenger RNA. Then based on the codes available with messenger RNA, the amino acids are provided by the transfer RNA because transfer RNA has the anticodon, and which will recognize the codon of messenger RNA. And similarly, this ribosomal RNA has a role because it forms the ribosome with protein. It forms nucleoprotein called ribosome, and that ribosome is the site of protein synthesis. So in this way, transfer RNA and ribosomal RNA help, and that's why we call them as housekeeping non-coding RNA. And there are certain non-coding RNA which regulate the gene expression, and they can be classified based on their length, either as long non-coding RNA or small non-coding RNA. If the length is more than 200 nucleotides, then it is long non-coding RNA, and if it is less than 200 nucleotides, it is called small non-coding RNA. And the small non-coding RNA play a major role in your gene expression and regulation. And some examples are microRNA, small nucleolar RNA, snRNA, small nuclear RNA, snRNA, small interfering RNA, siRNA, PIWI interacting RNA. This is a protein PV. So piRNA, which is of length 26 to 31 nucleotides. MicroRNA is of length of 22 nucleotides. So in this way, this is the we can classify the RNA. So coding, non-coding. Then non-coding can be divided into housekeeping and regulatory. Regulatory can be long or small. Then just we have discussed the functions. So functions of RNA, the major function is they act as code for proteins, and example is messenger RNA. And we can see how this messenger RNA, see, suppose this is a DNA strand, one strand is from 5' to 3' direction, and the other strand is 3' to 5' direction. So this is a double helix DNA structure. So if one is from 5' to 3' and the other is from 3' to 5'. So one strand is called the sense strand, the 5' to 3' direction strand is called sense strand, and the other strand is the antisense strand or template strand. And if you see there can be negative supercoiling of this DNA by the enzyme DNA gyrase, and we can get the two strands. And out of these two strands, the strand with 3' to 5' direction is called the antisense strand. This is the antisense strand, and this acts as the template for RNA synthesis. So by the action of the DNA dependent RNA polymerase enzyme, this transcription process occurs, and we get the complementary bases. So T for A for U, C for G, and accordingly in the 5' to 3' direction we get messenger RNA. So this is the messenger RNA, and 3 bases together form the code, so or codon, and the transfer RNA, which is similar to your T structure, it has the anticodon, and at one end this transfer RNA has the anticodon, and at the other end it has the amino acid. So AUG is the start codon, and the anticodon for AUG is UAC, and UAC at one end of t-RNA there is UAC anticodon, and the other end there is methionine, the amino acid. So most of the proteins have this methionine as the first amino acid. Then next 3 bases AUC stands for isoleucine, next 3 UCG for serine, and it continues until the stop codon comes. UAA is the stop codon. So the amino acids provided by transfer RNA will stop here because the stop codon arises, and in this case we get these 3 amino acids, so it will be a tripeptide: methionine, isoleucine, and serine. In this way, from DNA the peptides and proteins are synthesized, and RNA, the messenger RNA, has the same information as that of the sense strand. You can see here ATGAT, so AUGAU. Only instead of thymine of the coding strand or sense strand, we have now this U. Instead of thymine in the 5' 3' direction of DNA, that is the coding strand, we have U. The rest are same. So this is the function of messenger RNA. Then translation. The translation is the process in which based on the codon of the messenger RNA, the t-RNA provides the amino acid by recognizing through the corresponding anticodon. So this is the process of translation. This is translation. So that is another function of RNA. Here example is your transfer RNA. And as I discussed, the ribosomal RNA along with protein forms a nucleoprotein called ribosome. So we can say that ribosomal RNA is the catalytic component of ribosome, and it hosts the translation because ribosome is the site of protein synthesis. So this is the function of your ribosomal RNA. In addition to that, we have RNAs which regulate gene expression. So microRNA, siRNA, small interfering RNA, piRNA, these are all examples which regulate gene expression. One example, let us see microRNA. What actually it does, microRNA base pairs with the messenger RNA regions and target them for degrowth. If this is the messenger RNA, and the microRNA with complementary base will form pair, and it will be followed by the degradation of this messenger RNA. So there will be no translation and no protein synthesis. So in this way the microRNA can regulate. And there are certain RNA which can modify other RNAs, and examples are the small nuclear RNA and small nuclear RNA and small nuclear RNA. So this is all about RNA. Thank you.